Okay. You know what? Okay. So the objectives of this article were to assess and map the electrical conductivity as a measure of the soil salinity, and also to assess and map the soil total nitrogen, phosphorus, potassium, and sulfur variability. And this was done between both agricultural and fallow land, so land that was not cultivated for 5 years. And this was all done along a 90-kilometer distance gradient from the coast to inland in um, in the coastal region of Bangladesh. So the methods and materials. So this was done in the Noakhali Desert District, and they chose 4 different points. And are you able to see my mouse? Okay. And so each part right here is about 30 kilometers. And so they kept it even. And then soil samples were collected at depths of 0, 10, and 30 centimeters in the 4 different locations. And there was also distance. So it went from 0, 30, 60, 90 kilometers. And then they followed random sampling. And this was done, oh my God. This was done in and around the Noakhali district. So there was a bunch of different instruments and methods used. And so there was, in order to determine the different nutrients and the EC, so the Henna instrument was used for the soil salinity to determine the EC, and then the Walkley and Black oxidation method was used to determine the total nitrogen, and the Olsen method and Bray and Kurtz method were used to determine the phosphorus level, and it depended which method you'd use based on the pH. So if the pH was less than 7 or pH was greater than 7, and the spectrophotometer was used to measure both potassium and sulfur. So this study was heavily based on statistics. So ANOVA was the statistical technique used to determine the different results, and they analyzed all these results and put it into a table after they did all the statistical curves, and then the tables were all translated into a bunch of graphs. So I didn't really put any of the tables because they were very confusing, but I did put the graphs. And 2 important terms for the stats were the variance and the standard deviation. So the variance measured how far the set of numbers were spread out from the average, and then the standard deviation was a measure of the amount of the variance away from the mean. So now I'll be moving on to my results and discussion. So this shows the overall electrical conductivity for the agricultural versus the fallow land. And so there was a bunch of differences along the distance gradient. Between the agricultural and the fallow land, there was a higher value of the soil EC shown in the topsoil rather than deep in the soil. And then it was comparatively higher in the coastal area than in the mainland for both agricultural and fallow. And then for this figure, this was between the total nitrogen and the phosphorus. So again, we have the distance from the coast, and then we also have the different depth. And this showed a huge kind of difference, but there was a huge difference in matter of change, but then there wasn't a difference in the soil within the different like levels of the distance and the depth. But if you can see the soil samples that were collected from 0 centimeters and then 0 kilometers for the total nitrogen, and the 10 centimeter depth of the 30 kilometers and 60 kilometers. For the phosphorus, they were recorded for the highest value in agricultural land. And then for the next figure, there was an overall significant difference in the amount of potassium and sulfur. And so for example, like if you look at the 30 kilometer distance for both potassium and sulfur, and the 10 centimeter and 30 centimeter depths, which are of the 60 kilometers, then there was a significant difference between both agricultural and the fallow land. So conclusions. So there was an overall trend for the soil salinity, and so there was a higher salinity at the topsoil, and then a higher phosphorus concentration found in the coast, so closer than as opposed to the mainland. And then all of this data can be seen through the analysis of the variance, which they did through statistics. And some of the recommendations they had for this area in Bangladesh was strengthening polders, which is like the piece of land that's like low-lying land that can be, that's like taken away from a body of water. And for example, like by building dikes and canals. And then another thing that was said in the article was to wash the salts from the topsoil using fresh water, but you have to be careful with that. And then also another thing is just to have a bunch of crops that can handle all of that salt. And that is it. Any questions?
Good job, Rowan. Unfortunately, we don't have time for questions, but it's tough to go first, and you did a good job. So thank you.
Thank you.
Yeah, so you could stop sharing your screen, and we will move on to Isabella.
Can you guys see it?
Yeah, that's great. All right, so I'll introduce Isabella. All right, so Isabella Ancona is gonna provide a presentation to us that's entitled Geomorphological Transition Research for Affecting the Coastal Environment Due to the Volcanic Eruption of Anak Krakatau by Satellite Imagery.
All right. So this is what I'm going to be going over today. I'm just going to tell you about some background about Anak Krakatau, and then I'm going to be going over the objectives of the study, and then the methodology that they used, and then the results that they came up with, and then finally the conclusion. So Anak Krakatau is an island stratovolcano in the Krakatoa archipelago in Southeast Asia, which is situated between the island of Java to the east and Sumatra to the west. It emerged in around the 1930s and has been building up composite layers of lava and pyroclastic deposits with relatively frequent eruptive activity. And so the focus of this study is on the most recent eruption that occurred on December 22nd, 2018, with which triggered a large landslide on the southwest side of the island, as well as, and it also triggered other significant geomorphological changes. And the images here in figure 1 were obtained via SAR satellite imagery. And for future studies like this one, it's crucial to use SAR satellite imagery because it is not affected by atmospheric conditions, which may block the view of the volcano that you would like to study. And so just a little bit more background. Prior to the eruption in 2018, the geomorphological appearance of the Krakatoa volcanic complex consisted of a caldera wall, a volcanic cone formation, lava flows, and plain and coastal areas. Just to give you an idea of what it looked like before. And so the main objectives of the study were to monitor the geomorphological changes from the eruption in 2018 by using SAR satellite imagery and Sentinel-1 processing imagery. Additionally, the study calculated the total land area gained from the eruption, which we'll go into more detail in a little bit. And then this kind of study can be very beneficial in providing volcanic data for disaster mitigation, volcanic monitoring, and damage assessment in the event of an eruption. So the first thing that they did was they obtained SAR, or synthetic aperture radars, from the European Space Agency Sentinel application platform. And they were used to assess the major geological changes from the eruption. And so figure 2 is the satellite images that they obtained. So this top one is pre-eruption, and it was taken on December 19th, 2018. This middle one is post-eruption, which was taken on December 28th, 2018. And then this bottom one is current conditions at the time of this study, which was taken on February 17th, 2019. And then these images were processed and exported into ArcMap 10.4 for island boundary dimensions and land area calculations. So what they found, the first part of the results were the major geomorphological changes that they found. And so figure 3B here is post-eruption image, and it shows that the southwest side of the island collapsed. The crater collapsed, and this is where the landslide occurred. And then you can see here on the northeast and southeast side, there is some land gain compared to image 3A, which is pre-eruption. And then here down in 3C, for current conditions, there is still some land gain here on the northeast and southeast side, as well as around the volcanic crater. And those were the major geomorphological changes. And then for their calculations, this is a table showing the results for the pre-eruption, post-eruption, and current island area calculations. And all you really need to focus on is the image date, which is the pre, post, and current, and then this total land area, which is what they calculated. And they found that the land gain from pre-eruption to post-eruption was around 22,100 square meters, and then the total area gain from pre-eruption to the current conditions of the time of the study was about 150,300 square meters, which is quite significant. And so in conclusion, as we've seen, both post-eruption and current images show significant geomorphological changes on the Anak Krakatoa Island. Despite the land loss on the southwest side, comparison of pre-eruption, post-eruption, and current island dimensions further show that the island has been growing in size throughout recent volcanic activity. Anak Krakatoa remains active and is experiencing high levels of volcanic activity according to hazard reports from the Volcanological Survey of Indonesia. And as I stated before, this information presented today can be very useful to people like government officials to monitor volcanic activity and risk assessment and plan accordingly. Any questions?
We have time for one question.
I have a question. What made you want to do your presentation on this article?
I am really interested in volcanology and studying volcanoes, and so I feel like this is just one thing that I might be doing if I go into this field, I guess.
Awesome. It's really cool.
Great. Well, thank you very much, Isabella, and we'll move on to Emma. Okay, so it's my pleasure to introduce Emma Chantegros, and she's going to be talking to us about the transfer of organic pollutants from sewage sludge to earthworms and barley under field conditions. Go ahead, Emma. Emma, you're muted, unfortunately.
Okay, there we go. Can you give a thumbs up in the reactions if you think putting sewage sludge on crops to be beneficial? Oh, I see some. Okay, awesome. So I will get into the transfer of organic pollutants from sewage sludge to earthworms and barley under field conditions. There we go. I am going to talk about the background, objectives, materials, and methods, focusing on the results and discussions, and then ending with the conclusion. So what is sewage sludge? So they are solids separated from wastewater treatment plants. So when you go through a wastewater treatment plant, you have the water, which can be reused, and then you get this these solids. So often these solids were just put into landfills, and that took up a lot of valuable space in the landfills. So scientists decided to put it onto soil and crops to improve nutrients, mainly phosphorus and nitrogen, and also add soil organic matter, which has a lot of benefits to soil. But when you put the sewage through this wastewater treatment plant, the sewage sludge, you're left with organic contaminants. So organic contaminants can come from pharmaceutical products, detergents, and other chemicals kind of flushed down. And these aren't very harmful, but they do bioaccumulate, and long exposure can have negative effects on your body. So the four main organic contaminants that we're going to be focusing on today are galoxolide, triclosan, nonylphenol, or NP, and nonylphenol 1-ethioxalate. And so here on the image on the right, you can see the structures of galoxolide and triclosan, which are the two that we're going to be focusing on. So the objectives that we're going to be focusing on are determine to what extent the organic pollutants dissipate in soil and how much of these organic pollutants are transferred to the earthworms and soil. and the crops. So this study was done in southern Norway. And so what they did in a greenhouse is that they took soil that was used to grow oats, and they put it into pots. And this was a silty loam soil. And then they added sewage sludge. So they took sludge from 3 different wastewater treatment plants, so A, B, and C, and they mixed those with the soil. And so they had different amounts that they mixed in. So they did 20 tons per hectare and 60 tons per hectare. And then they added 2 adult earthworms and about 25 seeds of barley. And so, and they made sure that the levels of nitrogen were all equal, and as well as like they really specifically monitored evapotranspiration, so everything was very uniform. And then they also had a part of the experiment where they just did sludge and no barley. So sludge with soil and earthworms and no barley. And then so they dried the sludge, or dried the barley after it was grown, and they froze the earthworms to later like extract data. So the methods that they used to extract data was the Quetcher's method, which just stands for quality, efficiency, cheap, easy, and like quick. It's very simple, and the image on the right shows kind of the overall steps. So they took the dried soil and the earthworms and the barley grains to extract the organic contaminants. And then once they had those organic contaminants extracted, they used gas chromatography mass spectrometer to look at the concentration of those organic contaminants in each of the things they were analyzing. So here we have our first organic contaminant, galoxolide. And so there's 3 different colors, the red, the blue, and the green for sludge A, B, and C. And so we're going to be focusing on the sludge B. And so here we have the amounts of sludge that were mixed in the soil, 20 tons per hectare, 60 tons per hectare, and then this is the bare soil, so no barley. And then here we have day 0 compared to day 96, and these values are in nanograms of galoxolide per gram of dry weight soil. So as you can see, these are fairly high, and then these drop down after 96 days to fairly low. And these values are in the hundreds compared to triclosan, which is another organic contaminant that we'll be looking at. So again, we're focusing on sludge B, and here we have the different amounts of the sludge mixed into the soil, and we have day 0 of the amount of triclosan in nanograms per gram of dry weight soil compared to day 96. And as you can see, it's much higher, obviously, for the 60 tons per hectare, because you're adding a lot more of that sludge. And so these values, if I go back to this other graph, are way smaller. The triclosan is way smaller than the galoxolide. And so here we have a graph of the dissipation of organic contaminants. So again, just focusing on the 2, triclosan and galoxolide. And so we have the percent dissipation for each of the different amounts applied, and this is in tons per hectare. So the dissipation percentage for galoxolide was overall greater than the one for triclosan. And then we also have the standard deviation, which were measured when they they took the controls and they spiked it with the specific organic contaminants when they were analyzing their data. So that's how you can get these this standard deviation, which is how far is it from the mean. And then dissipation means how much the organic contaminants kind of spread and dispersed. And then here we have the concentrations of organic contaminants in the earthworms. So again, we have the different amounts of sludge mixed into the soil in tons per hectare, and the orange is galoxolide and blue is triclosan. And so these are the amounts in nanograms of contaminants per gram of earthworm. And so there was a lot more of the triclosan in the earthworm compared to the galoxolide. And then here you can see there's a large standard deviation from the mean, because this bar is fairly large. And then here's another image that I showed at the beginning, but this just kind of reinforces the idea of the difference between the amount of galoxolide and triclosan when there's uptake in worms when there's roots versus when there's bare soil. And the arrows show like the difference in amount of uptake of these organic compounds in the earthworms. And so in conclusion, so as we saw in the image before, the roots created different food for earthworms. So the reason why there was a difference in the amounts of organic contaminants in the worms of bare soil versus the ones with barley is because the roots were a different source of food for the earthworms, so they weren't just eating and going through the sludge. And then the concentrations of contaminants in barley grains are not significant. So that means that you can apply sewage sludge on crops such as barley to improve organic matter in your soil and increase nitrogen and phosphorus. However, you can't do this for all crops. One example is lettuce, because the leaves are so close, and the parts that you're eating it's just too close to the sludge to be like healthy. And the dissipation is substance dependent. So each organic contaminant, it just depends on like the chemistry of the molecule to like affect the dissipation. It doesn't matter on the sludge or anything like that. Any questions?
Fortunately, we don't have time for questions.
Okay.
But if you have questions for Emma, you can save it for the end. And if we have time at the end, then you can ask them. Thank you, Emma. Good job. All right, so it's Spencer's up next.
Am I a host yet? Or
You are, yeah, so you should be able to share your screen.
let's see. No, that's not it. Share application. That's not it. Okay, I just gotta share screen. Okay. My computer might be a little slow.
Okay.
Oh, shoot. Let's go to slide 1. All right.
All right. So it is my pleasure to introduce Spencer Chen, and his presentation is entitled Water Quality Assessment by Pollution Index Method in the Coastal Waters of Hebei Province in Western Bohai Sea, China.
Oh, I'm sorry to interrupt, but is my video, can you guys see me?
Let's see. No,
Okay. I wonder is because if I share my screen, my camera
camera won't work.
it won't work.
Okay, well, just go for it.
Okay, I'm sorry about that. All right. So I think we can all agree that pollutants have been affecting the waters of the world in a very negative way, to the point where it can reach dangerous levels for the environment and the people who need to use the water to survive. Popular areas, especially, just like the ones near the Bohai Sea, that will have an immediate effect to its environment. So I want to lead into the overview, which is we go over background, and then the objectives, materials and methods, which leads to discussion of results, and then ultimately the conclusion. So some background information is that the Bohai Sea is surrounded by land from 3 sides with poor water exchange. Major popular areas include the provinces of Liaoning, Hebei, Shandong, and the city of Tianjin, who actually kind of depend on this water. But the fact that since they're very popular city developments, this leads to a large amount of pollutants from industrial and municipal wastewater to be discharged into the sea. In addition, oil fields and large ports are distributed along the coast, which sometimes can cause accidents, and those oil spills can cause further damage to the natural environment. And we want to focus on pollutants such as COD, which is carbon oxygen demand, and phosphate as an immediate threat to the marine ecology. And they're analyzed based on the primary ocean productivity in the Bohai Sea. So some objectives are that we want to first look at the pollutant loads from point sources of 27 water stations that are going to be investigated along the coastal areas of the Hebei province. And we want to determine how impactful are the pollutants of the COD and phosphate concentration and how distributed they are. And then finally, we want to see if the organic pollution index and the eutrophication index can assess the water pollution level and water quality as good indicators and how well they can assess the conditions. And we want to particularly focus on the water conditions in the dry and flood seasons of 2006. So we move on to materials and methods. The first one is field data sampling. They're pretty important to water quality monitoring and assessment. One technique applied by one of the authors was to apply sediment data to investigate wind-induced sediment transports. More analysis have been made on sedimentations by other authors on estuaries near Asia-Pacific harbors. This data collected also provided information about the transportation of minerals, inorganic nutrients, and heavy metals over the last 3 decades. The next is the Organic Pollution Index, which is basically a comprehensive index that includes various water quality constituents. It takes accounts of the effects of the COD, dissolved inorganic nitrogen, dissolved inorganic phosphorus, and dissolved oxygen. And then by comparing with standard values and other pollutants with similar properties, the index could then assess organic matter in the aquatic environments. Oh, sorry. The next is the eutrophication index, which deals with eutrophication. And eutrophication is the over-enrichment of a water body with when it has too many nutrients, and then that results to accumulation of organic matter. This index takes account of the associated impacts of COD, dissolved inorganic nitrogen, and dissolved inorganic phosphorus based on the water quality, and is mainly used to summarize water quality monitoring data. So let's get right into the results and discussion. We want to be monitoring basically the terrestrial sewage in the Hebei province, which is broken up into two categories: the river outfall and industrial sewage that are directly being discharged into the Bohai Sea. There are around about 28 sewage outputs, but only 4 sewages are taken into account because they direct the most impact, which are the Shanghai Guan Development Zone and the Shanghai Guan Shipyard, the Beidahe West Treatment Plant, and the chemical industry of Sangyou. There are, oh sorry, there are six pollution source surveys carried out between April and October of 2006, and indicators show major pollutants such as fecal matter, ammonium nitrate, suspended matter, mercury, and so much more. The maximum concentration of COD, though, in most of the general sewages was mainly in April, and only parts occurred later on in the drier months. Maximum phosphate concentration also has similar results, along with suspended matter. This shows, the graph actually shows that the COD concentration is higher in the dry season than the flood season, which is similar to other local bodies. You can see on the left sides of flood season, and the red indications on both flood and dry are relatively the same because that's the most impacted area. But on the phosphate concentrations, it's relatively in the blue, which is low. But during the dry season, you can see the green all over the board, and it's like widely distributed of the pollution. In addition, the pollutant concentration is closely related to the flow rate runoff, which usually means a decrease in pollution with the increase of flow rate during the flood season. But sometimes it's opposite, and more pollutants can actually be carried off due to the increase. So what we have here is a graph of how the organic pollution index and eutrophication index are used to assess the percentage of polluted area by the percentage of stations. So from the graph, anything below the Roman numerals of 3 are considered relatively safe, and anything higher from 3 to 6 is considered the danger zone of heavily polluted areas. So the flood season, which is colored by the blue, is relatively safe and low in the 25% areas, and in the heavily polluted areas, it's even lower. But compared to the drier seasons, you can see a spike in the heavy polluted areas from of 3 to 6. And in addition to that, the organic pollution areas and eutrophication areas have a massive difference between the dry and flood season. Next, we have the spatial distribution of the organic index and how the eutrophication index for the flood and dry season of 2006. Organic pollution is high in the Qinhuangdao waters and near the Huanghar Harbor, which is where the red indication is for both flood and dry. Eutrophication is also pretty high in these areas specifically. However, during the dry season, there's a more widespread pollution through the water bodies, as seen before. So though there's concentrated on the left side of the graph, the left sides of both images, you can see some sort of concentration, but compared to the dry season, compared to the eutrophication of the dry season on the right-hand side of each image, there's more widespread, and the more widespread is, the more pollution you're going to get everywhere. So in conclusion, pollutant loads for industrial sewages are far more harmful to the environment than that of general sewages. From the 4 rivers back in the graph, there is a high concentration of COD and phosphate distribution, specifically at some points because of that industrial sewage. In addition, COD pollution is mainly from land point sources, such as the outfalls of rivers and sewages being directly discharged into a sea. Phosphate concentration is also mainly from the non-point sources from the coastal ocean. Generally, it is determined that the pollution level is actually much higher in the drier season than the flood season based on the data and the graphs. And at the time of this article, controlling pollutant emissions should actually be carried out based on the marine environmental capacity, which then should bring more attention to the distribution of pollutants during the dry season. And in the end, the pollution index method was considered to be useful and effective for the assessment of water quality in coastal environments. It can reasonably quantify the spatial distribution of pollution, which means they are definitely a reliable indicator. All right, any questions?
Unfortunately, again, we have run out of time for questions, but that was a good job. You can hold your questions till the end. Thank you, Spencer.
Mhm.
All right, so the next one is Camille Eaton, and I need to give you the option to share your screen. All right, there you go.
Can everyone see that where my presentation is? Cool. Just want to check.
Yep.
All right.
All right, ladies and gentlemen, it's my pleasure to introduce Camilla Eaton, who is going to present a study on the contamination of a calcareous soil by battery industrial waste.
Oh. All right. So by show of hands, how many of you use and operate battery operated systems? Just about all of you. Now, how many of you know what happens once those batteries are disposed of and what they do to a soil environment? All right, not as many of you. Well, in this study, that I'm presenting to you today seeks to address that question by analyzing the characteristics of soils that have been contaminated by battery waste. In this presentation, I will go over the background of where this experiment was conducted and what it sought to accomplish. Then I will move on to the materials and methods in which I will address the soil that was chosen and the various experiments that went into it. Then I will go over the results and discussion and address what data was collected and what it all means. And then in the conclusion, I will summarize what I talked about in the presentation, as well as talk about some future studies that could help enhance this particular study. So to begin, our experiment takes place in Quebec, Canada at an abandoned industrial battery site that was operated from 1913 to 1980. And essentially the objective of this experiment was to characterize this soil that had been contaminated by battery waste disposal by analyzing its particle size distribution, buffering capacity and pH, and heavy metal content and distribution. In order to conduct those experiments, they first had to collect the soil. So this soil was collected within the site at a depth to about 25 to 30 centimeters. The scientists involved in this experiment anticipated that the majority of the heavy metal ions would reside at the top surface of the soil, given that heavy metals do not typically percolate down into the soil profile with much ease. And then a control site was also established so that they could compare it with a native soil that hadn't been nearly as contaminated and therefore get an idea of what kind of effects this heavy metal contamination had. And then each of these soils were sieved via the ASTM method, in which a kilogram of soil was collected from both sites, oven dried, and then placed on a series of sieves. The sieve diameters range from less than 2 millimeters all the way to 5 millimeters. And then each of these were stacked one on top of the other and placed on a mechanical sieve shaker for about 10 minutes to ensure proper distribution. The buffering capacity was also measured in triplicate via a soil to water mixture ratio at 1:2, and then titrated with 0.5 molar nitric acid. And the reason they wanted to assess the buffer capacity is that the buffering capacity essentially tells them how well the soil will hold on to organic and inorganic constituents, in which the higher the buffer capacity, the greater likelihood it'll hold on to things like heavy metals and so on and so forth. pH was also recorded via two different methods, one with a soil to solution mixture ratio of DI water and the other with 1 molar potassium chloride at 1:2 ratios. And the reason they did this, they did two different methods, was so that they could actually calculate the point of zero salt affected charge. And you do that by subtracting the pH from the potassium chloride solution to the one with the DI water solution. And this will tell them the net charge of the soil. So in other words, if it's negative, it'll attract cations more likely and hasn't been as influenced by the metals that were placed on the soil. Or if it's, if it's positively charged, it'll attract anions and therefore has been heavily influenced by metal contamination. The heavy metal content distribution underwent a series of procedures also in triplicate. The first procedure was taking a gram of soil, extracting it with 25 milliliters of DI water, and then with calcium chloride solution to get the heavy metals that were bound to the water, that were water extractable. And the reason for this is that the reason they took or used calcium chloride solution is that calcium tends to be more concentrated in the soil as compared to something like magnesium. And so they felt calcium chloride was a much more representative solution than magnesium chloride. And then calcium also just acts as a better agent for getting the heavy metals to be water soluble. And so they chose to use calcium chloride solution as opposed to magnesium chloride solution for that reason. After this filtrate system, they then used that residue and extracted it with sodium acetate and brought it to a pH of 5.3 in order to separate the exchangeable heavy metal ions from those that were bound to the carbonates. And then that residue was then extracted with hydroxylamine hydrochloride in order to separate the carbonate-bound heavy metal ions from the ones that were attached to the poorly crystallized or amorphous iron and manganese oxides. And once again, that residue was extracted with hydrogen peroxide so as to oxidize the organic matter and separate the heavy metals that were attached to the iron and manganese oxides to those that were attached to the organic matter. And then lastly, that residue was digested with concentrated nitric acid to separate the organic matter-bound heavy metals from those that were attached to the silicates. And throughout each of these procedures, the concentrations of the heavy metals were taken using an inductively coupled plasma atomic spectrophotometer. So our first part of the results was the particle size distribution. And in this graph, you can see that there wasn't much change between the contaminated site and the control site. The only real differences that kind of start to occur visually are between the 2 millimeter diameter mark and the 5 millimeter diameter mark. However, when they use a soil textural triangle to compare the textural classes, they came out the exact same despite these percentage differences, and therefore particle size was deemed to not have too much effect inflicted upon them by the heavy metals. The buffering capacity of the soil was found to be exceptionally high, with 297 milliliters of nitric acid being required to change the pH from 7.6 to 7.4. The breaks in the buffering capacity are due to the neutralization of the heavy metal carbonates. The steeper incline towards the end of the graph is induced by the neutralization of lead carbonate, which has a lower solubility product compared to the other heavy metal carbonates. For the pH, it was found that the control and the contaminated didn't have too much of a difference in pH, with only a 0.3 difference. And then the point of zero salt effect was found to be negative for both the control and the contaminated, and the values were not too different from each other, indicating that it wasn't so heavily contaminated with heavy metals so as to inhibit its ability to attract metals and other cations. In terms of heavy metal content, interestingly enough, even though the control site was supposed to be used so as to not be nearly as heavy metal contaminated and to compare it to the contaminated site, both of these sites were well above the legal limits within Canada, with the exception of cadmium and the control concentration of nickel. The reason I separate lead in this particular graph is that the concentration of lead was significantly higher than the rest of the constituents. At the control site, it was found that there was a concentration of 1,970 milligrams per kilogram of soil. And then at the contaminated site, there was almost 25,000 milligrams per kilogram. And the legal limit in Canada is actually 50 milligrams of lead per kilogram of soil, therefore indicating that this control site was not far away enough or possibly had some contaminant issues unbeknownst to the scientists. In terms of their distribution, the heavy metals were found to largely be in insoluble forms. As you can see in these pie charts, the main colors that are standing out are the light green, light blue, the yellow, and the gray, indicating the residual organic, oxide, and carbonate forms of heavy metals, indicating to us that these metals were largely immobile and insoluble and therefore difficult to extract if that was to be the next step. So, in this study I presented to you today, we characterized the soils that were contaminated by battery waste disposal via analyzing particle size, buffering capacity and pH, and heavy metal content and distribution. However, what wasn't included in this study that would have been a really helpful addition is studies on contaminant persistence. This site hasn't been operated since 1980, and yet these heavy metal concentrations were still really high. And so I think, and given that the heavy metal concentrations were still so high, even after decades of not being used, it would really be helpful to see as to how long those heavy metals do persist in the soil and what long-term effects they have. And if you have any questions about that, I would be happy to answer them. Thank you.
Good job. Unfortunately, same situation. as before. So we'll press on. And Nicole is our next speaker. Thanks very much, Camille.
We'll just hit share screen.
Yeah. Yeah, Nicole, there you go.
Is it showing, Ronan?
Yep. Mhm. Okay, it's my pleasure to introduce Nicole Ermish, who is going to give us a presentation about fungal and bacterial rhizosphere microbiome associated with grapevine rootstock genotypes in mature and young vineyards.
Okay. So to begin, I will be going over the background, the objectives, materials and methods, and the results and discussions of this research. So essentially, this research article was trying to see if different plant types and genotypes of the same plant species have like partially different microbiomes in their rhizosphere. So they were trying to see if like specific bacteria and fungus were associated with different grapevine rootstocks. This study took place in Spain. They used one younger vineyard and one older, and within each vineyard, they had 5 grape stock genotypes within each. So the objectives of this research was to unravel the effects of rootstock genotype on microbial communities in the rhizosphere of grapevines, and to compare the relative abundances of sequence reads and DNA amount of Blackfoot disease pathogens. So the picture in the right of the little like log piece is what Blackfoot disease looks like, and it is the most common like pathogen that occurs in vineyards throughout the world. So they also are just trying to see if like the genotypes of different vineyards are affected by this pathogen differently. So again, they used 5 grapevine rootstock genotypes in 2 different vineyards, and they sampled this data over 2 years. They used a technique called like 16S rRNA gene and ITS high throughput amplicon sequencing, which essentially, after they extract the DNA from just their samples, they're using this method. It gets very complicated quickly, but this method to sequence those DNA segments and then identify like different bacteria and fungus through using this database that matches those sequences to like known bacteria. And then they used PCR, which stands for polymerase chain reaction. It's a method to like amplify DNA segments, and they use that to measure fungal pathogens associated with the Blackfoot disease. And then within their methods, they had 3 rhizosphere samples randomly collected per rootstock, therefore a total of 60 samples per vineyard. And when collecting these samples, they were going at depths between 40 and 50 centimeters deep, where the roots were denser and more complex, because that is an area where like more just like nutrients are released. And so they just wanted to like make sure they're accounting for like the ecosystem in that area. And then they, within their samples, they detached like the soil particles from the roots by using NaCl solution, and they centrifuged it and then discarded the top portion, and that remaining soil fraction is what they used for the DNA extraction. And so then within the results and discussions of this article, they found that there was specific bacteria and fungus associated with the different like genotypes of the grapevine rootstocks. They found that 82.9% of bacteria and 58.7% of fungus biota was shared between the vineyards. So they found that like there was not much differentiation between bacteria. However, there was quite a lot of differentiation in the fungus that they saw within the vineyards. And don't believe I mentioned this earlier, but the soils within the vineyards are the same, and they're not like necessarily far apart from each other, but it's just the difference of like a mature vineyard and a younger vineyard. So with that, they found that the genotypes in the mature vineyard had more of a differentiation within the microbiomes that were found of each genotype, whereas they did not find that with the younger vineyards. So yeah, like within each vineyard in the mature one, each different genotype had pretty different amounts or like different species found in the rhizosphere, whereas in the young vineyard they didn't. And they thought that one of the possible explanations to this would be because like younger vineyards, because they have more of just like a standard growth, and like the nutrients that they need are not as like specialized as in a mature vineyard. And so once a vineyard is at that mature stage, they require different nutrients is what they were saying. So they've also found that there was no correlation between disease resistances and the genotypes and fungal communities relative to the Blackfoot disease. But in past studies, they found that rootstocks that had tolerance to what is called ESCA and Petri pathogens, those same like rootstocks had less like Blackfoot disease in them. So they essentially are like, even though in this study didn't have enough evidence for like disease resistance with that, they found that genotypes that have a resistance to other pathogens have it like built in them to also resist Blackfoot disease. And yeah, that makes sense. And then, so here are two, like diagrams of the mature vineyard, and it is two different genotypes. It's only comparing two different ones within the mature one. And each bar represents like a different species of bacteria. And so it's just a visual representation of the difference in like bacterial species within a vineyard, but only just like slightly different genotypes of the vineyard. And so with that, this type of information is very useful for like discovering genes built into plants that have certain resistance to pathogens, and like being able to apply that then to other plants, and also the comparison of just whether or not like mature and young has any effect on just microbial communities, and just getting to know how plants affect their own rhizosphere, and being able to use that information for other species is really valuable. So with that, is there any questions?
Nicole, same situation as before, so we need to move on to Patrick's talk.
Thank you.
All right, let me know when I can share my screen.
Okay, you should be able to now.
Okay. There you go. Can you guys see that?
Yep.
Okay, cool.
Okay, it's my pleasure to introduce Patrick Fitzgibbons, and Fitzgibbon, and he is going to present a article that was about quantitative evidence for increasing forest fire severity in the Sierra Nevada and Southern Cascade Mountains, California, and Nevada, USA.
Alrighty, thank you, Dr. Appel, for that short and sweet introduction. Before I get into the actual presentation itself, I want to give a short little anecdote to explain why out of all the thousands of articles that I could have chosen to study, I picked this one in particular. So there's kind of two parts to this. First of all, when I was the first 4th of July I can remember, I was 6 years old. My dad got mad at me because I took the biggest firework we could have and I lit it just because I had such a fascination with fires. And then furthermore, this summer I'm actually interning with Crew 7, which is based out of SLO, which is a wildland firefighting internship this summer. So I wanted to be able to know kind of what am I getting myself into exactly. So there's that. And here's a little breakdown and overview of what I'm going to be covering with you guys today. I have those 5 different points, and then the information found within each of those points coincides with the information that was presented to me within the article. So, oh, and before I want to get into the more meaty part of my presentation, I just want to show you the area that was being actually studied for this study. So basically all of it is California. As you see, it's just the Sierra Nevadas, and a little bit is reaching into Nevada. And actually where I live right now, I'm about like 15 minutes from that western side boundary of the Sierra Nevadas. So it has some direct effect on me. So for my background, we've all heard the 5W questions, and I think that is a great way to describe what's going on, give some background. So who did this project? The 4 main authors, as you saw earlier, were the ones who pioneered this. And so what exactly were they studying? Basically, what they wanted to do was be able to take data and just make a quantifiable way of showing fire severity and size within the Sierra Nevadas. And this study, so when did it take place? It took place 1984 to 2006. And where it took place, I showed you guys earlier, basically from Bakersfield all the way up to the northern tip of the state. And I think the most important question when looking at this article is, you know, why would you want to study this? And what I found through their discussions and what the overall data showed was that it affects everyone, a fire in particular, and it like is coinciding with climate change, so it's almost escalating as time. Before this study, which concluded in 2006, there was no quantifiable maps or graphs showing that there actually was an increase in fire severity and fire size within California. There was some speculation from scientists, but there was no actual study done. So that was why this happened. So the objectives, I've already went over that first one a little bit. Basically, they wanted to be able to get actual data that I'll go into how they did that later, and be able to give a graphical representation to show people. And for this study, as I looked into it, they had a lot of trouble with data accuracy because you're dealing with, you know, a forest fire that's covering hectares, and it's not an easy thing to map. And especially since, you know, you're never in a forest fire, you can't really view it yourself, so you rely mostly on satellite imaging for that. And then there's multiple points of interest within this study that they focused on. Basically, it's the size of the fire that is going on in the Sierras, how severe that fire is, which is rated on a scale from low to high, which I'll get into more later. And then from those two different points, basically, can they make any trends or any lines of fit to go into the future? And before this article, there's the debate between whether forest fires were actually getting more severe and was a black and white issue. It was either a yes or a no, and you know, there's not a lot of reasoning behind those kind of arguments. And this article, I think, is unique because it kind of just gives the evidence, it gives the data, it gives the evidence, and that's kind of the extent of it, which is unique to me. And also the last, I think maybe most important point is the future applicability of this data to show, you know, maybe in 20 years, what kind of size fires are we looking for? Where are they going to happen? More continued objectives. I want to just give you guys a little look at one of the graphs that they had, and it looks a little complex, but I just want you guys to focus on the last two columns there. So the north and south on the x-axis, which just basically they took the region that they studied, cut it in half, and there was a north part and a south part. And then on the y-axis, it has percent of burned area. So using the three different categories of how severe fire is, basically they quantified what percent of the fire was, you know, low, moderate, or really high. And I thought the north-south data was really interesting because you obviously get a lot less water down south. So I would think, you know, you would have more severe fires there. I mean, there are more fires, but looking at this data, actually, that's not true at all. So moving to methods and materials, I cut this into two different parts because basically the data that they took to map out these fires and, you know, get the evidence that they needed they had to do it in two different ways. So first was the Landsat imagery, and basically this Landsat imagery is just a satellite that's operated by USGS and NASA, and it's said to be the most accurate when it comes to this kind of data that they were looking for. So that's what we used for that. And then moving forward, they had to have some in-field assessments of what was actually going on in the forests themselves. So they went into the field, and how they measured the severity was actually all in the field, and they would take a 30-meter radius circle basically, and then I believe every hectare or acre of a fire, I'm not completely sure on that, and then they would use the CBI protocol right there, which basically rates the kind of devastation that the fire did to the landscape with low, moderate, and high. And basically, I mean, it's self-explanatory. Low is, you know, not much has changed, the vegetation grew back quick, and then high being after a year, there's still complete mortality of the area. Now moving to the results and discussion. So this is a really scary looking graph, but basically what you just, to look at the data that is actually meaningful for this, you move to the x-axis and you look at 1980 forward for both graphs, because that from 1986 forward is when the data was actually being able to be taken in the field. And there's that increase you see on the y-axis. They're both in hectares, and then the x-axis is years. And it shows on the left side the total burned area. So basically you see that increasing slope moved from an average of 20 hectares to almost 50. And then you see a similar thing on the right side, which I thought that's very compelling evidence on itself. And you should be focusing on those little gray squares because that takes the average from the preceding 11 years and puts that number up, and you see it's just escalating at a pretty high rate. And then results and discussions continued. Basically, why does any of this matter? So there's a few different reasons. First off is water. When you have a devastating fire in an area, you get a lot of debris, a lot of ash, a lot of different chemicals and materials that kind of make that water supply less stable, less healthy to have. Second of all, habitat. In this study, actually, the second most prevalent area for a severe wildfire was where there was the highest percentage of carnivores, which was really interesting to me. So basically, these fires are wanting to happen in areas where there's a lot of animals, and the habitat for these kind of animals is already shrinking. So that's another reason. Fire hazards, I mean, that's obvious to everyone. Nobody wants their house to burn down. It's really dangerous. And the CO2 that gets released by a forest fire is almost like incalculable. I read somewhere that the forest fires itself release about the same CO2 as all like auto pollution, which is very interesting. So in conclusion, to wrap everything up, there's quantitative data now to show that not only are there more fires going on in California each year in the summer months, but the size increases, the severity is also increasing. And the main factor of why this is happening is because of poor forest management. The government only allows 6% of the forest to be burned that was being burned prior, like about 100 years ago. Like they shrank the percentage allowed to be burned to 6% of what it was, I believe, in like 1920. So that leads to a big increase in like fire fuel, and that is it. Any questions? But I'm sure I ran out of time.
Yeah, thank you, Patrick. We got to move on to the next one, and that is Brianna. So thank you. Okay, it's my pleasure to introduce Brianna Gonzalez, and her presentation is entitled Effects of Land Use Cover and Protection on Stream and Riparian Ecosystem Services and Biodiversity.
Thank you, Dr. Appel, for that introduction. So without further ado, let's get into it. Okay, so before I begin talking about the article itself, I'd just like to give you all a general overview of what's going to be talked about in this presentation. Like others, it's the introduction, starting off with the introduction and then going through the article and ending with the conclusion. So before I begin, I'd like to ask you all, what are some reasons as to why you believe protected areas such as national parks are important, or what do you think they contribute? And if you want to say it, you could put it in the chat if you want. Or actually, I can't see the chat, which is fine. Okay, I'm just gonna move on, but I'm assuming many of you thought about protected biodiversity, conserve natural functions and systems that occur, so also maybe even increased recreation because a lot of visitors go to these parks a year. And even though we have these assumptions and thoughts about what protected areas contribute, there's actually a gap between knowing the ecosystem services and biodiversity, differences between protected areas, non-protected areas, and whether or not the differences between those are very significant. So that was the purpose of this article, to kind of bridge that gap and to see whether or not ecosystem services and biodiversity are higher in a certain landscape versus another. So the study split the experiment into 4 different landscapes. So here we have protected forest, unprotected forest, unprotected forestry. Forestry meaning where timber companies occur, where there's the cutting of trees, and unprotected agriculture. So within these 4 landscapes, the ecosystem services above were being tested. And so the objectives of this article were to determine, once again, whether there are differences between the services and biodiversity in streams found in protected and unprotected watersheds, and also to determine whether or not land use and cover play a role in driving these differences. This study was based in Quebec, Canada, and the 5 ecosystem services were being tested across 28 different streams and 7 national parks. And one of the requirements for this study was that if a stream was going to be crossing an unprotected watershed, such as the forestry, and agriculture, the stream had to be at least 50 kilometers from that national park or preserved or protected area, that they're being compared to. The next thing is how these different services were assessed. So there were 5 ecosystem services plus biodiversity that were being assessed across these 4 different landscapes. Water quality was assessed by the concentration of benthic invertebrae and also phosphorus and nitrogen concentrations in the water. Carbon storage was assessed through the amount of tree species and abundance of these species throughout the terrestrial environment. Wild foods was assessed based on the abundance of 5 different types. And wild foods, an example of a wild food is like raspberries. Recreation was tested by the amount of trails that were available and open to the public and if they were used. Habitat quality was actually assessed using an index provided by the Canadian government. And the Canadian government, to make this index, the government combined aquatic biodiversity and terrestrial biodiversity standards and the environmental conditions that they believed are needed to support species in both ecosystems. And lastly, aquatic biodiversity was again tested by the amount of benthic invertebrate that were concentrated or that were in the water, and terrestrial biodiversity was based on the amount of tree species that were around and present. The models used for this experiment are as shown. GLMs and ANOVAs are mixed models that are used to, or they were used to assess the ecosystem services and biodiversity and how they varied across all those 4 different landscapes. The Kendall-Rank correlations, those were more assessing interactions between the ecosystem services and biodiversity and determine whether, or demonstrated whether or not a certain ecosystem or an indicator or a biodiversity indicator created a change within another, and if that change also occurred kind of reverse, like if they were dependent on each other basically. And the PermaNova was used to compare all ecosystem services and biodiversity and to test whether or not the differences between all 4 landscapes was as significant as believed to be. So based on this graph, you could see that there wasn't a significant difference of the ecosystem services provided by protected forests and forests, but compared to forestry and agriculture, there was a significant difference. And here, these images just kind of take all the information that was gathered and put it into an image. So a few of the highlights that I just like to point out is that surprisingly, benthic invertebrate concentrations were the highest in unprotected watersheds such as agriculture, but yet the lowest in forestry. Ecosystem services were seen to be higher in all around in protected forests. The terrestrial biodiversity was the lowest in agriculture. And that sorry, I lost my train of thought. Okay. And also, or lastly, another highlight was that phosphorus and nitrogen concentrations were the highest in agricultural watersheds. So back to this, I'd just like to point out that the main conclusion or result that came from this article was that there wasn't much of a difference between protected forests and forest. It's as if the protected title had no real role in determining the differences between these ecosystems, which was very interesting because we all usually believe that protected forests and parks maintain the most biodiversity, better water, and things of that nature. So even though that isn't the case, having a protected title as guaranteeing all these services, it's still one thing to consider that if we were to get rid of that title, land use and land cover can undergo so much change that can possibly put these ecosystem services and biodiversity indicators at risk. So in conclusion, one of the highlights was that the key findings were that protected streams and riparian zones of all different landscapes can provide ecosystem services and biodiversity, each in their own way. Even though protected areas did, on average, have a higher amount of ecosystem services and biodiversity indicators recorded, the differences amongst all four landscapes wasn't as cut and dry, which was very interesting because that kind of disputed, or at least disputed my assumption of national parks. And one of the reasons why they think this could be true is because of regulations and laws. So let's say for agriculture, one of the reasons why they might have had lower ratings is because farmers are usually rarely lenient with the trees that are growing past the protected area. So that being said, there's less trees available to, and roots or root systems to kind of filter out the nutrients being used, like phosphorus and nitrogen that are being used on the soil. So without that filtering process, all those nutrients are making their way into the streams. So one thing that I didn't really see with this article, or they didn't really consider, was seasonal variability. So that is one thing that I would, if I were to make a suggestion to this, whoever did the study, or if I were to do this in my own way, I'd kind of take that into account, because I'm very sure that seasonal variability, meaning temperature, precipitation, and things of that nature, could affect the recordings of the services and the biodiversity available. The message that I'd like to leave with, the message that I'd like to leave with you all is that regardless of what a landscape looks like, or what you think, or what you perceive, each landscape provides its own ecosystem services and contributions, regardless of what its appearance is. So that being taken into consideration, I think the public, like ourselves, and also people like land developers, need to take that into account when creating buildings or planning to make a change to the land, just to take that into account and see and determine whether or not their actions are going to have an effect on these landscapes and services. Thank you, that is it. Any questions?
Thank you, Brianna. Unfortunately, we got to move on to Liam's. So Liam, you're up next.
All right. You guys see my screen?
Yes.
Oh my God, I can't see it. What the heck? Wait, sorry.
That's all right. All right, ladies and gentlemen, here's Liam Graham, and he's going to talk to us about a paper that was entitled Observations of Static Coulomb Stress Triggering of the November 2011 M5.7 magnitude 5.7 Oklahoma earthquake sequence.
Great, thank you so much, Dr. Pell. First, I'll start out with the overview. So I'll be going over the objectives and the introduction, some background, data collection, static Coulomb fault failure, and my conclusion. So first, I'd like to start off by asking a question. Does anybody think that humans have the capability to trigger an earthquake? Just raise your hand. Okay, so a couple of you raised your hand. Well, in this paper, well, to give some background, we usually expect earthquakes to be in subduction zones or deep underwater near mid-oceanic ridges, but sometimes rare events happen and we get earthquakes in the middle of a continental plate, like in the center of the United States in Oklahoma. So the objective of this paper of the study is asking, do fluid injection wells trigger seismic activity in or near faults in central Oklahoma area? Okay. If you're unfamiliar with what fluid injection is, it's the process of drilling deep into the ground about 2 kilometers and cracking the earth and injecting it with a mixture of water, sand or mud, and chemicals to extract natural gas or oil from the center or from deep in the earth for human consumption. So moving on to the data collection. So in this paper, there were 3 major earthquakes. The first one, I'll be calling these event A, B, and C. As you can see labeled, if you can see my mouse, and they're labeled as stars. And event A was the initial earthquake on November 5th, 2011. And then this was followed by, in the same 24 hours, another earthquake of 5.7. And then on November 8th, another earthquake of magnitude 5. And they were able to record 110 different earthquakes. Oh, sorry about that. 110 different earthquakes, which are labeled with black dots. And these were the most clear. They have clear P wave arrivals and SP ratios. So if you've ever seen a seismometer, it has a lot of movement, and clear P wave arrivals don't have too much shaking in it, and it was clear to them to be able to get data from it. And they collected data from different stations like the University of Oklahoma, EarthScope TA, the United States Geologic Services, and Netquakes. And I thought the EarthScope TA is pretty cool because it's an array of seismometers that's all across the United States. And that also includes Alaska and Hawaii. So moving on. So now we're going to move on to the static Coulomb fault failure. And this is how they figured out whether or not these earthquakes were triggered by fluid injection or by some other outside source. So just to clarify what this equation means, because it looks like a lot of gibberish, but here we see the change in shear stress. It's a combination of change in shear stress, friction coefficient, normal stress, and pore space. And if you guys are confused on what shear stress and normal stress is, shear stress is when 2 faults, if you can see me in the screen, drag against each other. And normal stress is stress put on something that from all sides that's always there. So yeah, so also this this equation gives results in units of bar, which are units of pressure. And when the static Coulomb fault failure is positive, they concluded that that is evidence of fluid injection. And when it's negative, it's evidence of an external pressure. But then they found out that once fluid injection, here, wait, sorry, my screen's not, what the heck? Okay, so back to this image, we can see these black dots. These are 110 earthquakes that they recorded. And they found out that event A and B, these 2 right here, which were the 2 largest earthquakes that occurred, were both had positive Coulomb fault failures, indicating that they were caused by fluid injection. But C did not had a negative Coulomb number, so they were curious on why that happened, and they soon found out that the extra stress that caused event C was caused by the other earthquakes prior. So yeah, and then here is a diagram of all the earthquakes. So this represents the focal, all the little beach balls that you see on the screen are called focal mechanisms, and this shows the direction of stress or compression and stretch in the earthquake, and this allows you to see what type of earthquake it is. So if you can see the black section and the white sections of the beach balls, also known as focal mechanisms, the black section is compression and the white section is being stretched. So that just indicates that it is a right lateral earthquake or fault. And the black focal mechanisms represents the positive Coulomb numbers, and then the gray represents the negative Coulomb numbers from the results. And then the pink or magenta focal mechanisms are anomalous, and they were unsure what happened with those earthquakes. Also, yeah. Okay, and then in conclusion, they were able to determine the static Coulomb stress changes due to the event A were consistent with the triggering of event B. So this suggests that fluid injection did trigger events in the nearby faults and cause earthquakes in Oklahoma. And a takeaway that I would like to say for this is there's a positive and a negative. So I would say the positive takeaway for this is we can retrieve data from these earthquakes for geological purposes, like figuring out what's beneath the earth and figuring out what's in our crust or what's in the continental crust. But the negatives are also human risk and damaging buildings and stuff like that due to earthquakes. But yeah, I wanted to make my presentation kind of short, just so I can get some questions. So does anybody have any questions for me?
Time for one. Liam, I thought it was very interesting.
Thank you.
Yeah, thank you. All right, so we will move on to Josh.
Can you guys see my screen?
Yep.
All right.
So it's my pleasure to introduce Josh Hedden, who is presenting on a paper entitled The Impact of Wildfire on Soil Organic Carbon Cycling in a Mixed Hardwood Evergreen Forest. Take it away, Josh.
All right, thank you, Dr. Appel. Did you know 4 of the 10 largest fire years on record have occurred in the past 15 years, burning over 2 million acres in each occurrence, accounting for billions of dollars in relief and rescue funds? But what you don't hear a lot about of these fires is how it affects the soil and the carbon cycle. All right, today I'll be going through the background of just how important the carbon cycle is. Then I will reference the 2 objectives of the research article. I will then go on to talk about the experiments that took place and how they're set up, followed by the data collection of these experiments and what the data may mean, and finally finish up my presentation by answering the question, why do we care? All right, for a little bit of background, I'll be going through just how important the carbon cycle is, as forest fires play a substantial role in the global carbon cycle as a whole, with forest loss and degradation due to fire accounting for an estimate 12% of total CO2 emissions around the world, while 12% is recorded of above ground emissions. Globally, soil organic carbon, which is 3 times the size of above ground carbon, is unfortunately not recorded. And since major disturbances such as fires have on below ground carbon cycles can only increase these uncertainties of how much carbon is actually being released. So for my 2 objectives today, the first will be, how does fire change the distribution of soil organic material among density fractions and depths? And the second is, how does fire affect the general character of soil organic material with varying carbon to nitrogen ratios? For this experiment, soil samples were taken from Oregon's Siskiyou National Forest in 2004. Just 2 years prior, there was a 200,000 hectare biscuit forest fire. Sites were selected so that variation was taken into account, so that age, aspect, slope, and elevation were minimized. Samples were taken from 1 of 4 hectare sites. For each had burn severity classes of unburned, low severity, mixed severity, and high severity, and were tested for root, fragment, rock fragment, carbon and nitrogen concentration measurements. An additional 2 plots were sampled for high severity burn sites and unburned sites. 2 to 20 soil cores were taken from each site at depths of 0 to 5 centimeters, and then again at 5 to 15 centimeters using an auger. Once soil samples were collected, volumetric rock fragment contents were calculated using estimates of soil volume and rock volumes, while organic carbon and total nitrogen concentrations were determined using a CNS 2000 microanalyzer, and root densities were calculated using a pneumatic pump. And finally, density separations were determined as soils were sieved before being placed in an oven overnight and then weighed. As you can see from this table, soil organic carbon concentrations did not vary significantly with burn severity, but soil N concentrations in soils from burn stands were significantly higher than that of nitrogen concentrations from the control study. These high severity burn stands were more than twice that of the control sites. And as for the variation in carbon to nitrogen ratios, they followed the variation of the nitrogen concentrations, as control plots had significantly higher carbon to nitrogen ratios than that of soils from the severely burned sites. As you can see in this graph of rock percentages, the difference in rock fragment content among soils were varied from soil from high severity burn plots that showed that they are 4 times greater than that of the control plot, as well as rock fragments from low and mixed severity plots were also 3 times greater than the control. The higher erosion assumed that as soil fine particles are removed from the soil surface by that of wind or water erosion, it left more coarse rock fragments to be left behind. And qualitatively, these rock fragments were more than the less eroded control plots. As for the root graph, While looking at this route table, you can see that forest floor measurements were significantly reduced in all burn stands, and the reductions were highest in high severity burn areas. As for areas that experienced high severity burning, it also shows changes in forest floor composition, with evidence that woody debris in the litter layer was nearly double that of unburned control stands. Qualitatively, fine root abundance seemed to increase with increasing burn severity, while medium root abundance seemed to decrease with increasing burn severity. All right, in conclusion, carbon concentrations were seemingly unaffected by fire severity, while nitrogen rates doubled with increasing severity, leading to carbon to nitrogen ratios to increase for the control plots. Rock fragments and fine root abundances increased with fire severity, while medium root abundances decreased. And finally, if I had one take-home note, it would be that high severity fires are increasing almost every year and leading to more carbon in the atmosphere and seriously eroding soils, leading to barren soil patches and hydrophobic soil conditions that can lead to worse disasters such as landslides. And this concludes my presentation. Thank you.
Thank you, Josh. Similar story as the other ones. We need to move on. It's Catherine Jansen's turn.
All right.
All right, ladies and gentlemen, it's my pleasure to introduce Catherine Jansen, whose talk today is entitled Intense Human Pressure is Widespread Across Terrestrial Vertebrate Ranges.
Okay, thank you, Dr. Appel. I chose this topic because not only am I very interested in biodiversity, but biodiversity loss is considered one of the biggest things that humans are currently having an impact on. So in this talk, I'm going to go over the background of why the authors decided to do this research and why it's important, the objectives, the methods, results, and just kind of the take-home message for this. Okay. So as I'm sure everybody here knows, that humans are having a huge impact on the world and the environment, and throughout Earth's history, there have been 5 mass extinctions, and many scientists now agree that we're in the 6th mass extinction right now, largely due to anthropogenic causes. Currently, we're at risk of losing over 1 million species, and one of the main pressures on this is land use change, usually for agriculture. And it's just incredibly important that we preserve this biodiversity because it affects ecosystem function, which in turn affects everything. And the research kind of got started because of a initiative from the United Nations called the Decade on Biodiversity. And so for the past 10 years, they've been encouraging research in regards to how, threats on biodiversity and how we can protect it. One of the main goal of this is to halt and stop extinctions. The objectives for this research, one, they wanted to focus on terrestrial taxa. So this includes birds, mammals, amphibians, and reptiles. And they specifically picked species that have been assessed for distribution plus their extinction rate or risk. And they want to quantify the number of species ranges under intense human pressures and investigate the extent of these pressures across a couple of different scales. And those include pressures on taxonomic classes, species level of endangerment, and species range size. And then they looked at data over course of time starting in 1993 and ending in 2009. Okay. So since the researchers wanted to kind of process this on a global scale, they used more of just synthesis methods to kind of compile this information. They combine existing species distribution data along with spatial data on human pressure. And they got the human pressure data from human footprint maps, which already exist and have assigned values based on 8 different human pressures and determine high versus low risk areas across the world. The 8 different pressures that are assessed in this are built environments, population density, electric infrastructure, croplands, pasturelands, roads, railways, and waterways. And with this information, they calculated proportions of species ranges in each particular human footprint index so they could assign values, and mostly they wanted to just determine the species vulnerability. And so the results of this, which I guess aren't shocking, sadly, they found that 85% of species have over 50% of their range exposed to intense human pressures, and another 16% have absolutely no portion of their range that isn't affected by human pressures. And species with smaller ranges have a larger overlap with this, so they're even more at risk of losing potential range size and therefore having a higher risk of extinction. And then in regards to the time frame, since 1993, species entirely exposed is up by 44%. And this is a graph from the research, and it breaks down species ranges by particular taxa. So we have amphibians, mammals, birds, and then all threatened species here. And the gray bars indicate like the total known range of species, and then the red bars are adjusted to include or to exclude the areas under intense human pressures. So kind of as you can see, all of the species were negatively impacted by these pressures. Everyone is losing potential range size, particularly the threatened species, and that includes critical, endangered, and vulnerable species. Some of them are losing virtually all of their habitat range, which is only going to increase the likelihood that they're going to become extinct. And yeah, just every species is affected by this. So in the discussion, like these are just key elements that we have for determining extinction risk. And with this information, hopefully we can continue to identify species that need to be protected, maybe some that are in more high risk areas versus low risk areas. And it's just every species can be affected by this. And while some will be able to persist in these conditions, they're still not going to be free of consequences. They're going to have a smaller range size, and therefore probably going to lose numbers as well. One of the things I did find interesting was, and they noted this, that there were a couple, well, just one main problem, and it's that it didn't add in every pressure that has an effect on biodiversity. Some of the pressures left out were anthropogenic climate change, pollution, infectious disease, invasive species, overexploitation. And while it would be really difficult to include all of these in addition to the factors that were included, just for further research, if we're going to build on this, we can try to include a couple of these different factors in addition to everything that was researched here. And also more specifics on individual species like winners and losers and who's going to be more or less at risk. Okay. And in conclusion, we just really need to prioritize safeguarding biodiversity. It's critical for everything on the planet. Ecosystem functions affect everyone, starting on a microscopic level all the way up to elephants and bigger species. And hopefully with this particular research, we'll be able to target specific habitats and either do more restoration efforts or put them up for protection and conservation efforts. And in addition, we can use the human footprint to assess more species vulnerability. So we can focus on specific species or categories of species, such as amphibians, which are going to be affected at the highest rate. And if we can do this and we can work together to hopefully minimize our impacts or protect species ranges, hopefully we can prevent further mass extinctions. And that's all.
Thank you, Catherine. So we are like right on time or slightly over. If anyone would like to just hang in there for a little bit and ask questions of the presenters who came before them or came today, that would be just fine. If not, then I'll see you all next week. And just remember to do your peer review for the cover letter and resume. There's a rubric online that you can fill on that's available to Canvas, and upload your evaluations of your peers to the appropriate OneDrive folder. And I'll make sure that you all have access to it in the next few minutes.
Did you say that? Oh, sorry.
No, go ahead.
I was going to ask, did you say that the scoring portion of the peer review wasn't nearly as pertinent as like the comment section? So we weren't able to like fill out our, okay.
Yeah, so like Camille said, the comments part is the more critical part for these peer reviews, for the presentations that people gave today. And if there's certain things that you'd like to score, then you can go ahead and do that. It's not critical that you add up the points either. Just that you provide some feedback, because I think that what the speakers will find helpful are trends. And then the other thing is that everything from today was recorded, so you can view yourself, and I would encourage you to do that, because I think that there's a lot of constructive things that can come out of that. Any other questions?
Hey Chip, I was just wondering if I could talk to you about cover letter and resume stuff after.
Sure.